আচ্ছা ওই অ্যাটেন্ডেন্সটা দিয়ে দাও। হ্যালো, গিভ এর অ্যাটেন্ডেন্স। আচ্ছা লাস্ট ক্লাস মানে স্টারডে আই আপলোড ওয়ান কি বলে মিউটেশন ইন ডিটেলস ওয়েতে, ঠিক আছে? ডিটেলস ওয়ে, the details, if you are not understand then we discuss about it. Okay, I discuss it elaborately, but in the details way or molecular basis of the mutation that will be discussed in the previous class as upload in the in your Google Classroom. Today we discuss about the regulation of gene expression in case of prokaryotic. ঠিক আছে, আমি ক্লিয়ার? বুঝতে পেরেছো? হ্যালো, দেখতে পাচ্ছ? হ্যালো, টু সিন?
হ্যাঁ স্যার দেখতে পাচ্ছি।
লাস্ট ক্লাস ওর যে পিপিটি molecular basis of gene expression that will attach your Google Classroom. Okay, you can see. And today we discuss about the regulation of gene expression in case of prokaryotic. So before we study the regulation of gene expression of prokaryotic, what is gene? Obviously we are know that what is gene is the functional unit of genome, or specially it's called the transcribed mRNA is called the gene. The DNA molecule, the chemical vehicle and hereditary, it is the chromosomal functional unit of life. It is called the gene, and namely the genetic information content in the cell, all the genetic information content in the cell, especially for the DNA. And the bacterium Escherichia coli, which is one single chromosome content around 4400 single genes. In case of mammalian or human genomes, around 23 pair of chromosomes, it contains around 30,000 to 40,000 genes. So actually gene is nothing but a protein expression, or gene expression is nothing but the protein expression. One gene produce one protein. One gene responsible for synthesis of or expression of one protein. That means gene expression is nothing but the protein expression. This gene expression, but how it will be regulated? Actually ধরো suppose in case of suppose one organ will be damaged due to some accidental, at that condition their growth hormone will be increased, and their particular gene will be stimulated, and the gene expression will be occurred, and ultimately that type of protein which is help for recovery or repair of the growth, the repair of the particular damaged portion. And at that time the growth hormone is which are used as a factors to aggravate or to stimulate the gene expression or to protein synthesis. তার মানে here the growth hormone used as a inducer. So we can see study and what are the types of দেখো introduction and repression, inducer and co-repressor, type of regulation of gene expression, transcriptional regulation, and the Operon model, and translational control and post-transcriptional regulation. What is the gene expression? And the gene expression is the process by which the information encoded in the gene in used to direct assembly of the protein molecule. That means the gene is nothing but a mRNA. That means mRNA contains some specific sequence. That sequence expressed as a form of amino acid or a form of polypeptide. That means a mRNA which produces a particular sequence, and that particular sequence, a particular amino acid sequence, ultimately you get a particular protein, and which is applicable after the post translation modification. That's why it is called the gene expression. Gene মানে একটা compound, it is a transcribe or it is a information that will be expressed. When it will be expressed, but the expression depend on some regulatory mechanism, either the positive or is the negative mechanism, or some genes are inducible, and they are divided in the basically the gene are divided in the three types: inducible, repressible. So gene expression means gene is expressed. ধরো সাপোজ ইউর এক্সপ্রেশন, তোমাকে কিছু বলা হলো, ইউ এক্সপ্রেস, তার মানে ইউ টেল সামথিং, ধরো ইউ টেল মি ইউর সেলফ, তো ইউ এক্সপ্রেস ইউর সেলফ. That's why when the gene will be expressed, that means a particular protein ultimately the gene will be exposed. Gene will be exposed means the gene will be stimulate the protein expression. But how it will be expressed, but when it will be expressed, that will be dependent upon the some agent, or that is called the inducer, or they will be, and at that time it is called the inducible. When sometimes it will be stop it, or to regulate a down regulation, that is called the repressor. So generally, when it is needed, then gene will be expressed. When not needed, then automatically gene will be not expressed. But there is a two controller. One is the inducer, another is the repressor molecule. And what is the regulation of the gene expression? That is, first of all, the gene expression, right? I say it. Then is the how to regulation. Obviously, continue if the continue gene express, their body will be increases. Their body cell mass will be increases. So there is a problem. So there is a everything must be regulation, either increase or the decrease. The regulation of the gene expression is include as a wide range of mechanism that are used by the cell to increase or decrease in the production of the specific gene products or protein or the RNA. That is called the regulation. I mean, the expression, when it is expressed or not, that is called the regulation of gene expression. But this gene regulation of gene expression in case of prokaryotic, it's much more easier process. But in case of prokaryotic, eukaryotic, it is a very, very tough and very, very complicated procedure by which the gene expression can be regulated. In case of eukaryotic, only the 2-3 single genes are regulated, or total 4,500 single gene. In case of humans, here the 40,000 gene are regulated, and how which gene is express, which gene is the stop. So very, very tough procedure and how it will be regulated, and the various types of mechanism involved to regulation of gene expression. Gene expression in case of prokaryotic, here the very simple, the DNA molecule, it produces mRNA, and mRNA is a growing amino acid chain. In case of eukaryotic, the nucleus, transcriptional process, cytoplasm, mRNA, and transport, and finally this protein will become. So ultimately gene expression is nothing but a protein expression. But when protein will be expressed, actually protein will be synthesized. That is the protein synthesis, which is discussed in the previous class. What are the translation process? Basically, when the gene will be expressed, at that time the protein will be synthesized. But how it will be synthesized, and when it will be synthesized, and how it will be regulated, which is the requirements, that is the initiation factors, or that is the initiation factor. And when initiation is processed, then the elongation started, and after that, when the elongation is completed, then it will be stopped. Then it is called the termination process. Otherwise it will be, unless it will be going on, ultimately it will be a very big compounds, and that will be not desirable compounds. So it will be must be a stop. Every process is not continued, any other process. Okay. So what the type of gene expression? One is the constitutive gene expression. It is essentially gene for the living cell. Constitutive gene expression. What is the constitutive gene? Or it is called the housekeeping gene, or it is basically it is called the housekeeping gene. Housekeeping genes means there is a constant expression. The constant expression of the gene, the product or the protein of a gene is required all the times in the cell. That means constantly they are expressed. These are called constitutive genes. Therefore, the constitutive gene for the housekeeping gene and express at more or less constant rate at most all the cell, and therefore there is no subject to regulation as per enzymes of citric acid cycle. In case of citric acid cycle, the 10 processes involved, 10 enzymes are involved. Either it is slow or it is the higher rate, but it is a continuous process. This is called the constitutive gene expression, and especially for the live process, they are continuously expressed either in the low rate or in the high rate. That's why it is called the housekeeping gene. Just in your body, in your building or in the house, the one housekeeper to maintain your house. That is called the housekeeping gene. Another is the inducible gene expression. Enzyme is catabolic function. Inducible means there is a inducer. That means to aggravate the or increase the gene expression by the agents or inducer. One example, enzyme in catabolic function. Suppose glucose breakdown, glycolysis, TCA cycle, vintage phosphorus. They are basically called the inducible gene expression. And what is the down regulation repressible gene expression? That means the down regulation. The enzyme is anabolic pathway, that is called the repressible. Repressor and depressor, another term is very common. Depressor and repressor, basically they are both are the same. The repressor gene expression means their enzyme is the anabolic, there is some growth or new formation, that is called the, or generally that the slowest process, but comparatively than the inducible gene expression. So gene expression that will be classified in the graphically. In case of inducible gene expression, or dekho, it will be started here. Will start the activity of the enzymes involving the lactose utilization. Here will start, then the log phase, then this just type of microbial growth. So here will be started it, and then that after times 5 to 10 minutes, it will be immediately then the continuous phase. This is the types of gene expression. It is called the inducible gene expression. In case of lactose, it is a inducer molecule. In case of prokaryotics, in case of lactose operon, here is the lactose is a inducer agent who increase the this type of, or it is called the A type of gene expression or inducible gene expression. That means here when the inducer will come, the gene will be expressed initially slow, then the fast, and then after that they are constantly gene expression. In case of the repressor gene expression, they are generally the tryptophan operon. Here is the Lac Operon. Here is the tryptophan operon. It is basically the repressor molecule. Here the tryptophan will be added, and the gene expression will slow down and or down regulated. They go initially down and then fast or rapidly down it, then they slow down the process and ultimately slow down this process. Now, same types of phenomena, just like a reverse of the inducible and the repressible gene expression. Induction and the repression. What is induction and what is repression? In case of the E. coli, Escherichia coli synthesis for the beta galactosidase of the enzymes, details in discussion the Lac Operon systems. The means for the hydrolysis for the lactose in the glucose and galactose, and studied in the considerable details as early in 1960. Lactose or galactose and the enzymes beta glucose and galactose, and such enzyme which are synthesis which can be inducible by adding substance known as the inducible enzymes. This beta galactosidase, which galactosidase beta lactose is converted to glucose plus galactose. Glucose plus galactose. কোথায় গেল? Actually, lactose is a disaccharide. In presence of galactose, glucose plus galactose. Here it is shifted here. Actually, glucose plus galactose. Glucose, lactose is a disaccharide. In presence of beta galactosidase, it is a inducible enzymes to convert catabolic function. I last discuss about here. It is a catabolic function. देखो, inducible gene or the enzymes is catabolic function. That means breakdown process. Here, lactose is converted to glucose plus galactose is a monosaccharide in presence of the beta galactosidase. So this is called the inducible gene expression, or it is the gene inducible enzymes. Such enzymes, those are synthesized, can be restricted by adding end product of the corresponding biosynthetic pathway, or by who knows the repressible enzymes, and this genetic system would be known as the repressible system. Obviously, this beta galactosidase, if they continue, that immediately continue, the glucose will be breakdown. But when with the excess amount of the glucose, what types of, in that case, the repressor molecule come and the depression molecule down regulate the or catabolic process will be reduced. Inducer and the co-repressor. The substance, those are addition. Induced synthesis is enzyme is called the inducer. And the end product, those, that means end product means here is the glucose and the galactose. Those addition with restrict the synthesis of biosynthesis of enzyme is known as the co-repressor. Although Ordinarily, only the substance acts as a inducer, but rarely they are molecules that adequately resemble with the natural inducer, but are not the metabolized themselves in the enzymes. So co-repressor, which means the inducer molecule along with the co-repressor, that means they control the inducing effect of. In an inducible system, the active repressor plus inducer, the active repressor molecule, actually inducer molecule to induce the gene expression. But when the active repressor bind with this, suppose you কোনো একটা কাজ করতে চাইছো, at that time তোমার বাবা বাধা দিল, at that time your father is stop the functions, that is there is a inactive the repressor molecule. In an repressible system, inactive repressor, here is the active repressor. In an repressible systems, in an inducible system, here is the repressible system. Inactive repressor, here is the active repressor inducer, inactive repressor. No, actually repressor is active, but inducer also active, but repressor function is not. That's why the inactive repressor will be function will be reduced, ultimately inducer function will become. Inactive repressor and co-repressor, the active repressor. Inactive repressor plus co-repressor, ultimately active repressor মানে down regulation, switch off. Type of regulation of the gene expression, one is the positive regulation, another is the negative regulation. When the gene express, your gene expression will be increased by the some agents or inducer agent, that is called the positive. When the expression of the genetic information is quantitatively increased by the presence of the specific regulatory element, it is called the positive regulation, and this agent is called the inducing agent or inducer. Here is the positive regulation, the one is the promoter and operator, ultimately the transcription and activator, that is the inducer molecule. Here is the activator bind with the operator, then promoter will express it and the transcription will become there is then the translation will become. The element modulating the positive regulation is known as the activator or positive regulator. Next is the negative regulator. When the expression of genetic information diminished by the presence of the specific regulatory element, it is called the negative regulation. This process is called the negative regulation. Here though only the repressor molecule, here the activator molecule, here is the repressor molecule to bind with the operator, then the promoter will be stop out this, the transcription will be stopped. Here the transcription will start, here the stop transcription will not occur due to negative regulation by the presence of repressor molecule. So the element or the molecule mediating the negative regulation is said to be a repressor molecule, or no repressor is required. And transcriptional regulation, in case of the Operon model, the friend Jacob and the Monads in the 1961, on the basis of their study, the inducible system for the synthesis of beta galactosidase enzymes of the E. coli, proposed the model to formation of induction as well as the repression of enzymes and synthesis, the model popularly known as the Operon model. In case of Operon, there are two Operon models we are studying, different types of only the Lac Operon, another the tryptophan Operon. This Operon model sum as the presence and the unit is called the Operon. And Operon is nothing but a the Operon concept is a coordinate unit of genetic expression. Actually, a different types of gene coordinate simultaneously. This is called the Operon system. What is the Operon system? And the Operon genes which are controlled onto the activity of the protein synthesis or structural genes. The Operon consists of Operon model. The operon consists of the two types of gene. One is the operator gene, another is the structural gene. Here you can see it. This is called the structural gene and this is called the operator gene or regulator. It consists of three elements. One is the regulator, promoter and the operator. And here is the three structural gene in case of Lac Operon. ZYA, beta galactosidase, beta Y is the permease and acetylase. Here is the three enzymes will become. One is the beta galactosidase, Y is the beta permease and A is the acetylase. And another is the regulatory gene. One is the regulator, promoter and the operator. How it will be controlled? This operator genes and the structural gene we discuss. Thank. Any question regarding this up to this? Because this model will start, then it will be stop, completely discuss. Otherwise, it will be a problem. So next day we discuss about the Lac Operon system. Totally only discussed it based on the Lac Operon. If any problem up to this, what type of gene and what is the gene regulation, what are the types of gene regulation, I think this is the very preliminary molecules, preliminary terminology. You can remember it. Otherwise, this Lac Operon or the tryptophan operon. Then the next class we discuss about the Lac Operon and tryptophan operon. Okay.